Boom. Okie dokie, kokie, kokie, kokie. Can everybody hear me? You might notice, just a quick sound check, you might notice I've decided until I get my lighting sorted, instead of looking like a white marble, I've got a hat on. So there we go. Just make sure you can hear me. Yep, 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 yep. Hello, hello, hello. Nice, nice, nice. Listen, I decided to go a bit earlier. I was going to do a poll, but yeah, there's only a few viewers anyway. So to see what people prefer 6 p.m. or 7 p.m. But I decided to go 6 p.m. today and see if people prefer that. Anywho, we're talking about insects today and fish and their respiratory systems. So that's what came out on top of the poll ahead of aerobic respiration. So that's what we're going to go through today. So, what is an insect? Does anybody know what an insect is? What makes an insect an insect? How many body parts does an insect have? How many legs? Well, an insect has got 6 legs, 3 body parts: head, abdomen, and thorax, the upper part, abdomen, and a head, of course. And they've got an exoskeleton, a hard shell on the outside made of chitin or chitin, however you like to pronounce that. And that is or is one of the substances in their exoskeleton, and that is surrounding them. Yeah, they are an invertebrate. First ones to evolve on the earth, actually. And the first animals to take flight 400 million years ago, these guys evolved. They evolved before trees. So before there were any trees on the earth, there were insects. Of course, there were other plant forms in the sea before the insects, but the insects evolved a long time ago. So they're a highly successful group of animals. Yeah, first animals to take flight. So before all the flying dinosaurs that are now extinct, you had the insects. All right, let's move. We need to go through how do insects respire, and essentially, I mean, I've got a PowerPoint, right? And the issue is sometimes it's clicky clicky on the PowerPoint. You're used to that at school, click 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 click. Sometimes I think it's not worth it. I could just sit here and tell you everything you need to know in 5 minutes on an insect. But we go through the PowerPoint. Huh? Okay, so let's go through the PowerPoint. So there's only really 7 or 8 things you've got to know about the insect's respiratory system in total, and that's all you need. You could fit it all on one revision card for insects. So anywho, let's go through. Ooh, look, there's another one. That's nice and colourful. Okay, so I'm going to go through. First bit that you need to know about for the insect's respiratory system are the tiny holes on the side of the insect. Yes, but it's also in A2. Of course, this will appear in A2 as well, but you are right. Yeah. So it is in AS biology, but it will appear in A2 questions as well. So what are the tiny holes? They're a bit like stomata on a plant because they can open and close. Spiracles. Lovely, lovely, lovely. Awesome. Well done. Yes, of course. These are called spiracles. So well done. It's one of about the 8 things you've got to know. So there's not a lot to know. Here's the rest of the respiratory system of an insect. And this is literally it. So if you can in the chat, see if you can put down what is A, what is B, what's C, what's D. There's really only 3 bits there. A, B, and C. So why don't you go for it? See if you can write down A, B, and C. And that's all there is to the insect's respiratory system, essentially. Ah, Matilda, if we were dealing with the humans, I would give you a high five because that would be the bronchioles. But in insects, they've got a slightly different name. You're absolutely right. That you could get a question comparing the mammalian respiratory system, i.e., the lungs, to an insect, and they would be the bronchioles in a human or a mammal, but not in an insect. You are right. The trachea for B. Spiracles A, trachea B. What do we call those tiny little tubes, very, very small tubes that don't have any strengthening? A, I believe, I forgot. That's the spiracle. You already named that one. That's the spiracle A. B is the trachea and C is the tracheole and D is the cells. Yeah, the body cells, the stoma or the somatic cells. Sorry, not stoma. The somatic cells, the soma of yeah. So we got the body cells. So how does this work? Right, the spiracle can open or close. Why would it close? To reduce water loss. So there might be times where they want to reduce water loss, a hot day, dry day, so they can open and close these. Oxygen literally diffuses in. The insect relies on diffusion. and the oxygen literally diffuses down these tubes called the trachea. These are strengthened with rings of a material. Our trachea is strengthened with cartilage. What are the rings? What is the substance that is found in the trachea of an insect? Yeah, it's chitin or chitin, however you like to pronounce it. Brilliant. So we've got these rings. What are they for? They're for support, just like our trachea, our cartilage in our trachea, our rings of cartilage. So same thing. These are very small tubes. They branch off extensively. They have chitin in them. Then they branch down to the tracheoles. And the tracheoles, we want to talk about the fact that they have a large, extensive network of tubes that gives them a large surface area, which increases the rate of diffusion. Now, insects rely on the fact that these cells here are respiring and using oxygen for aerobic respiration, creating an oxygen deficit here and a steep concentration gradient from outside the spiracle all the way down to the cells. And because insects are small, if an insect was my size, this wouldn't work. Simple diffusion alone would not allow the oxygen to get there quick enough. But due to their size, this is a brilliant respiratory system. The oxygen diffuses down a steep concentration gradient all the way. And the beauty of the tracheoles is some of them actually go into cells through the cell membrane and right next to the mitochondria. So they're literally like injecting the oxygen into the mitochondria. So it's a very rapid diffusion of oxygen into the body cells of the insect. And this enables them to churn out ATP at a serious rate. Okay, they produce ATP at a really, really high rate. And that's why when you've tried to swat a fly and you're like, oh my God, does this thing not tire? They don't get tired. Whizzing about like that, they can produce so much ATP. Not as efficient if they were our size. There are no insects our size, unfortunately. So yeah, that's all you really got to know about insects and their respiratory system. There's one more bit on insects and then we'll do some questions. So one more little bit on insects. So this is literally it. Spiracle, tiny holes. And the spiracle is not found in the head, but the thorax, which is the upper part of the body, and the six pack, the abdomen. Okay, and basically, they don't have a six pack, but yeah. The oxygen diffuses in through the spiracle, down the trachea, along the tiny tubes that are strengthened with chitin, you could be asked about that, through the tracheoles, which create the large surface area, and into the body cells. Now, there is some fluid. There is a little tiny bit of liquid. Most of these tubes are empty and filled with air. Okay, so it's mostly gaseous diffusion down there, but there is a little bit of fluid at the end of the tracheoles. What does fluid do? It increases the gases dissolve and it kind of increases the contact area. It kind of increases the surface area, having a little bit of fluid at the end of the tracheole. So it actually helps with diffusion. Now, one more thing that we need to learn about for this, before I give you a couple of questions and then we'll move on to the fish. One more thing that you need to learn about is how do insects ventilate themselves? Does anybody know? Yes, you're right, Oscar. Fluid moves out by osmosis as the lactic acid builds up. That's absolutely right. So the buildup of lactic acid, sometimes when they're really exercising hard, they produce a bit of lactic acid near the end of the tracheoles. And what happens is you get a bit of osmosis into the lactic acid and a little creation of fluid there at the end of the tracheoles. Yep. Yeah, that's right. That's right. Yeah, you've got abdominal pumping. Now, what does this look like? So, abdominal pumping. Let's have a little look at some of that. This is not what they're talking about, okay? That's not abdominal pumping. This is abdominal pumping. Are you ready? Um, so, this insect is working out its six pack, doing some crunches, some sit ups, and essentially, you'll see the vibration. This insect is pumping in, ventilating the spiracles with air by creating a pressure gradient. So totally different to how humans do it. We do it by changing, remember, we do it by changing the size of our thorax by increasing the volume, decreasing the pressure, and then down a pressure gradient. They're using abdominal pumping. They can pump their wings as well. It's not just the abdomen. You need to know they can pump their wings. Let's do a couple of questiones just to test you guys up on this. So, okay, let's have a little look at this one. No alveoli, Matilde. No alveoli. They have no alveoli. And why don't they have alveoli? Because Now, I don't want to say insects don't have any blood. They have a circulatory system. They have a open circulatory system, so that's where there is a fluid that is pumped through their kind of cells, in between their cells, and that is not to deliver oxygen. So the delivery of oxygen goes through the trachea. Each spiracle is connected to a separate trachea, so they have many trachea, and those trachea branch, so they are branched, but then they branch even further into tubes without kind of chitin, and the tubes without chitin are the tracheoles, and they go directly into cells, and or near to cells, or even into cells and into the cytoplasm and near mitochondria, the tracheoles, so they can directly deliver oxygen straight to the mitochondria at very short diffusion distance because they're small, and a large surface area. Yes, so A is the spiracle, guys. I know it looks weird. What are you going to guess that is? You've only got a couple of guesses. And then B, this is a tube that has rings in it or lines going along it. So it tells us this is part of an insect's gas exchange system. We've only got three answers. We've got spiracle, we've got trachea, and we've got tracheoles. We know that the holes are spiracles. Does that look like a hole? Well, I guess you could say it does. And then you should know that this has to be the trachea, because it has chitinous rings. You can see those lines, they are chitin. So yes, that is the correct answer. So you've got to use a little bit of knowledge. Spiracle and trachea. Yeah, lovely. Hi, hello, hello. Yep, awesome. Okay, let's have a little look at another question. So let's go, let's go, let's go. So on that, got a couple of questions here. This one I didn't talk about because it's part of the circulatory system rather than the gas exchange system. This is not part of the gas exchange system, but you might want to be able to answer this. So we've got an aquatic crustacean, Daphnia magna. Now, if you do Ed, actually, I don't know if it's in AQA or OCR, but it's definitely in EdXL Salter's A-level biology. You have to do a core practical where you look at the heart rate of the Daphnia and you give them caffeine. I can't remember if that's in AQA and OCR as well. But you have, yeah, hemolymph. This is the blood-like liquid, but it's not designed to deliver oxygen. It's just amino acids and sugars and nutrients. So that's hemolymph. Right. Question 16, this one here. Which of the statements A to D describes the circulatory system of an insect, of a Daphnia magna? What do you think? Yeah, so insects, what kind of circulatory system do they have? Now, their respiratory system has tubes. This is just to get you away from this. The circulatory system is about how they deliver nutrients, how their blood moves through their body. Their blood is not carried in tubes, so they have an open system, and it's single and open. So there we go. Yeah, single, but it's not in tubes. Okay, the gases are in tubes. So I imagine originally, and again, this is imagination, I'm no insect expert, but I imagine in evolutionary terms, insects probably originally had something akin to hemoglobin that would move through their body carrying oxygen. And then a later on adaptation was this whole spiracle and tube system to deliver oxygen directly into the cells. Or maybe one came before the other. I'm not sure the order, but they are two separate systems. So their circulatory system's main design is not to deliver oxygen. It does have some protein-like hemoglobin proteins that can carry oxygen, but that probably does like 5% of its oxygen carrying. Most of its oxygen is through the tracheal system, which is its respiratory system. Right. What about this one here? What do you reckon? A, B, C, D. Which one is the spiracle? I mean, look at that diagram. That diagram is foul. What do you reckon? If you had to eliminate, spiracles are holes on the thorax and the abdomen. So what one could be a spiracle? What are we going with? C? We going with B? It's really hard to see probably on your screen, but B is kind of pointing to one of the tubes. So it's either going to be tracheoles or trachea. It's going to be here at C. is the only one you can really go for. D looks like it's where they excrete. A is somewhere in the head. B looks like part of the tracheal system, the tubes. So I would go with C. It's on the outer side of the body. So C is the correct answer. A-level chem is on Mondays at 7 p.m. 7 p.m. Mondays. I might change to 6 p.m., but I'll see. But A-level chem is 6, 7 p.m. on Mondays. Right, 8. The diagram shows part of the gas exchange system of an insect. Which of the labels over here is the trachea? God, I don't even know what D is. D is some weird sack. I don't know enough about the insect's respiratory system outside of the A-level knowledge as to what you got to know. So we're looking for chitin. We're looking for evidence of chitin, or we're looking for it to be connected to a spiracle. So Matilda asked earlier, is there only one trachea? No, these are all trachea. They all have chitinous rings. They are trachea, all of these, or chitinous, however you want to say it. And then down here, look, the tracheoles that go straight to the muscle cells do not have chitin and are smaller. So that's how you're going to know. Awesome. Okay. Right, let's move on. Right, you guys, just a quick summary then. Adaptations of the insect's respiratory system. So just a quick reminder, spiracles can open and close to prevent water loss. The tracheal system provides a large surface area by having an extensive network of tubes, trachea and tracheoles that branch out. Large surface area means rapid diffusion. Some of these tracheoles have a direct path into cells to deliver oxygen by simple diffusion down a concentration gradient into respiring cells where the oxygen concentration will be low. What else can we talk about? We can talk about rhythmic pumping to ventilate the spiracles with fresh air, or beating wings to do that as well. We would also expect all the same things. What are the 3 adaptations of all exchange surfaces? Whether it's your villi in your small intestine, whether it's the alveoli in the lungs, what do all exchange surfaces have? Thin walls. So I would expect there to be thin walls. You've got that large blood supply, of course, all of that business. So all of those same things you're going to get in every exchange surface. So short diffusion pathways, all of that business. Right, let me give you a couple more questions, then we'll move on. We can talk about prevention of water loss. So yes, you can talk about spiracles closing to prevent water loss when the air is dry or extremely hot. Yes. Okay. Yeah, they are very much like stomata. Okay, folks, see if you can do this question. And then what we'll do is we will move on to do the old fish, which is a little bit extra. I do need to talk about the insect practical. It's literally 3 bullet points to remind yourselves. So you could fit everything you need to know about insects could go on the back of one revision card. So it's literally spiracles, literally trachea with chitin, and then tracheoles, and then rhythmic pumping of the abdomen or wings to ventilate the tracheal system. So what do you think the function of the chitin is? This is some insect that has it's a honeybee, and it has a parasite which is labeled with M. So these look like fleas or something. Some sort of parasite that has invaded the breathing tubes of the insect, the trachea. What's the function of C? Maintain stability. Yeah, support. Absolutement. So maintain stability and support. The mites use their mouth parts to bite through the walls of the trachea. They then feed off the hemolymph. So they open up the tubes and then suck in the fluids that are being pumped by the insect's heart through its system. The blood-like liquid that bathes the cells and organs of the honeybee suggests one other way in which the presence of the mites might affect the honeybee. How else might the fact that there's these parasites living in those tracheal tubes affect the honeybee? Yeah, not nice, is it? I suppose humans, we get fleas and things like that. and all sorts worms, God knows what other parasites. But the honeybee gets these living inside its tracheal system. Okay, so what would be your justification? You're right though, yeah, less oxygen transferred to the cells, maybe because the honeybee's respiring, and maybe the liquid will, maybe if the liquid fills the tracheal tubes, this blood like hemolymph, it will disrupt the diffusion of oxygen, all sorts of things like that. Maybe some oxygen will leak out. There's all sorts of things that could happen. Let's have a look what the mark scheme says. You'll get all the different options, so you can just have a little think. Their presence restricts airflow, so literally resists the flow of air. Yeah, leakage of hemolymph deprives the tissue cells of oxygen, nutrients, amino acids, etc. The parasites use oxygen. Yeah, or it says disease transmission. Okay, I thought it said you couldn't mention infections or whatever. All right. This is one of the, just for your own interest, this is a giant dragonfly, 70 centimeters across. We know that during the, I think it was the Carboniferous era, that insects could grow like big like that. Scorpions could get this big, like, and dragonflies could be almost a meter wide. And that was because the atmosphere had 35% oxygen. So you had a steeper concentration gradient, which allowed the oxygen to move more swiftly into their cells, and they could make more ATP to sustain a large size. Nowadays, the amount of oxygen in the atmosphere, it is theorized, is not enough to sustain insects above a small size. So even though I say their respiratory system's pretty cool, it can't sustain large insects. Okay, what else? What else? Oh yeah, dissection. So if you get a revision card and you write all the bits you got to know, which is only a few bits, the other thing you want about, you don't want to get into an exam without knowing what you'd say about an insect dissection. I would literally just have 3 bullet points. Pin the insect to a board, take off some exoskeleton with scissors, whatever, snip it off with a scalpel, with scissors, and then inject saline solution into the insect. Saline solution is like what you clean your eyes out with. It's like eye drops. Basically, it's like water with some salts and some other very dilute sort of stuff in there. So you inject that into the insect. If you then take a little slither, a little sample, and you look at it under, you can actually peel it out and then look at the trachea under the microscope, and you'll see the silvery tubes, apparently. And apparently you can see the rings of cartilage as well. So, uh, yeah, well, the carbon dioxide, I would go with, yeah, yeah, you're kind of right that, but I would go with like they're using up the oxygen, right? So it's going to get less oxygen because they're respiring as well and using up oxygen. Um, but yeah, you could talk about it. CO2 levels rising might cause issues. So just make sure you know about insect dissection. So it's literally pin it to a wooden board, snip off a bit of the exoskeleton, jab a needle in there, inject some salt solution, some saline solution, sorry, in there, and then you can peel off the trachea, look at them under a microscope, and they'll appear as silvery tubes, and you can even identify the chitin. Right, we want to move on to fish. But before we move on to fish, just to remind yourself, 3 ways in which the insect's tracheal system is adapted for efficient gas exchange. Now this says the word explain, so you need to say what it's got and how that helps it. Gas exchange, Mr. Editor. Insect dissection comes in AQA and OCR that you have to know about dissecting insects. For AQA and OCR, you got to know about how to dissect insects. So if you're doing OCRA or AQA, you got to know about dissecting insects. For EdXL Salters, I don't think they do insect dissection. Mainly A level today, but I will be doing, I might be starting some GCSE ones on the on a day. I might put up a little, I'm going to put up a little text poll of what day would be good for GCSE with some options. Yeah, so you want to talk about the branching of the tracheals, extensive network, large surface area, maybe fluid at the end of the tracheals, increases the contact, increases surface area. Spiracles to allow oxygen to diffuse into the insect. I don't know if you'd be able to say rhythmic pumping. Is that part of their exchange system? I don't. I don't think that's part of the tracheal system. That's a behavior, the rhythmic pumping, isn't it? And the wing beating that ventilates the tracheal system. So I don't know if they will have that. You never know. They might have it on the mark scheme. I've got the mark scheme right here, but which I'm going to put up in 10 seconds. So just be thinking, what other things would you say? Some of the tracheoles go directly into the cells, so you've got a short diffusion distance. We've said large surface area, and I wonder whether they'll give us anything about, well, you can't say large capillary network because they don't have a closed circulatory system, so they don't have capillaries. Um, they have hemolymph. Right, here are your answers, folks. Any three of these? Tracheoles have thin walls, so the thin walls, they love that. Short diffusion distance to cells, highly branched number, so short diffusion distance. You could also say highly branched, so large surface area. So look, lots of them end in the same thing. Trachea provide tubes full of air, so fast diffusion. Fluid in the end of the tracheoles, faster diffusion. Fluid at the end of the tracheoles increases the surface area again. So many times saying increase the surface area and short diffusion pathway or short diffusion distance. And look, they have include body can be moved, muscles to move air, so maintains diffusion concentration gradient. So they're saying that rhythmic ab workout they do, those sit-ups that the insects do, remember, what they're saying is that this business, they're saying that this is also part of the tracheal system. Personally, I wouldn't have said that was. I was surprised, but it was on the mark scheme, so we'll go with that. Right, let's move on. More insect questions? I think we're done with insects. Let's move on to the fish. We're doing insects and we are doing fish today, the respiratory system. And next week we'll do something else. I put up a poll every week and people can vote on exactly what they want to go over. So, yeah. And the poll this week, aerobic respiration was beaten into second place. So it was going to be Krebs cycle and all of that. But it was beaten into second place by people that wanted the fish gills, fish, counter current flow, all that business, and the insects. So we've basically covered everything with insects. Um, oh yeah, we can do a live stream. I'll put that up on the text poll and see if people want to do it. Control of high heart rate via the sympathetic and parasympathetic system. Yeah, we can do that. We can do that. We can do that. We can do that. Noradrenaline and all that business. Okay, acetylcholine. So we're going to go on to fish. I have more insect questions maybe at the end, but I want to do. I've got some more exam questions on insects at the end, but I want to go through this. So let's go. Okay, straight off, if you compare, now we're not doing the circulatory system, but I thought just a quick reminder on the circulatory system of fish. So you could get this kind of question where you have to know about the animals, a mammal circulatory system. What's the difference between the circulation of blood in a fish and in a mammal? You can use the words open, single, double, closed as a clue. Mr. Editor, don't worry about dropping £100. I don't need the money, but I will do a GCSE, and I'll do that on. I'm going to put up a text poll. Actually, I need to find out when GCSE kids want to do it, because I don't know the times and the days where they'll want to do it. So I'll put up a little text poll. Yeah. So mammal has a double. Fish, you can see, has a single. How do we know it's a single? Check it out. The blood comes back from the body into the heart, out the heart, goes to the gills, picks up oxygen, becomes oxygenated, and then it still has enough pressure to make it round the fish, back to the body, and back to the heart. So it's a single circulatory system, not like humans, which is in, out, in, out. It's just in and out, right? So it's a single circulatory system. A similarity is that they're both closed. They're both inside tubes. So that's a similarity. What's another difference? Check out the heart, guys. Check out the structure of the heart. What's the difference in the structure of a fish's heart to a human heart? You might be able to spot another difference there. So we might be able to see, yes, you could say blood goes to gills and not to lungs. Yeah, I guess you could say that the blood goes to the gills instead of the lungs in humans. Also, the heart has just one ventricle. It looks like one big open ventricle, which just one big fat ventricle. We have two ventricles. So there we go. So one ventricle. So a different amount of chambers. Yes, Slay Queen. We could talk about the chambers. It does have some chambers. It has a ventricle right there, but we have a left and right ventricle. There's just one ventricle here. So there we go. Right. Let's get onto the gas exchange system. So, like the insect, where we said with the insect, you had to know, just running through the insect again before we finish, or before we go through fish. So we had spiracle, we had trachea with chitin, then we had tracheoles directly into the cells, and we had the rhythmic abdominal pumping to ventilate the system. Simple as that. The fish, you've got a couple of other key words, but they do the same thing. It's how do they make gas exchange work? They have something called gill filaments, and they have lamellae. Now, gill filaments are sometimes called primary lamellae, depending on your exam board and the exam question. Most of the time, they're just called gill filaments. You're allowed to call them either. So I call them gill filaments. Some people call them primary lamellae. They then further subdivide. So you have the gill, and then you have these structures coming off of the gills called gill filaments. And then on top of each gill filament are like, if you imagine you chopped a plate in half, are little plates called gill plates or lamellae, depending on what you want to call them. So you've got gill filaments or primary lamellae, and then you have gill plates or secondary lamellae. I just call them gill filaments and lamellae. Anyhow, the lamellae are smaller than the gill filaments, and both of these structures, generally you only have to mention the name of one, because it will be what gives the fish gills a large surface area, and you can just say the gill filaments and lamellae, or you can just say one of them normally on the mark schemes. Okay? Gonna briefly go through. The fish have an interesting thing called a countercurrent flow system, but we're going to look at what's called a parallel flow system and what would happen if they had a parallel flow system. So, excuse my crusty drawing, and I know my face is over some of the writing, don't worry about that. Imagine this is one of the gills. Okay, so this is the gill filament, gill plate, and you've got the blood running across the gill in this direction and the water running across the gill. These little circles here are oxygen. Which way is the oxygen going to diffuse? Well, it's going to go from the water into the blood. Simple pimple, right? Like this. So from a high concentration to a low concentration. So imagine in comes the blood, in comes the water. They run across the gill plate, and the water is moving across the gill plate into the blood. Lovely. We do have an issue. Look at the oxygen is diffusing in. What's going to happen if they're running together and all the oxygen has moved over into the blood? What's our problem with this? What's the problem with this parallel flow system? So imagine this, the blood is running, the oxygen has already diffused across. We got an issue. This oxygen that's here is now running parallel to water that has given up its oxygen. If anything, it's going to want to move back. So this causes a problem. A parallel flow system will not fully work because now the blood is high in oxygen. By the time we get to the end of the gill plate, the blood is high in oxygen and the water's low in oxygen. You're going to lose some oxygen by diffusing back out into the water before it's expelled from the fish. So, OMG, we have this issue. How are we going to make sure that the oxygen doesn't start diffusing back? And how are we going to make sure that the oxygen fully goes into the blood and we get maximal uptake of oxygen? This is the crusty parallel system, and this is the issue. If you see this graph, this shows you the limitation of a parallel system. So imagine this is the length of the gill. So the blood comes in, the water comes in. The water's high in oxygen, the blood is low, it's deoxygenated. It's just come back from the body. We saw that circulatory system of the fish. So it's delivered all its oxygen for respiration. As the water and the blood are going next to each other, the oxygen moves into the blood. But then we get to a stage where they're 50-50. Look, 50%, 50%. We get no more net diffusion of oxygen. So the maximum saturation of the blood in a parallel system is 50%. So actually, the fish can't rely on that. So what the fish has evolved to happen is that the blood hits the start of the gill plate here, and the water, fully oxygenated water, hits the other end of the gill plate. So they're going past each other like that. And what this means is that you always have fresh water which is high in oxygen going past blood that is low in oxygen. And the way we want to describe this is called a counter current flow system. So it's called the counter current flow system, and what it basically ensures is that along the entire length of the gill plate, the oxygen is always moving from the water into the blood down a steep concentration gradient from high oxygen in the water to low oxygen in the blood. And that's all you got to memorize for this. So even if you find that hard to understand, you can just memorize a sentence and then you've got it nailed. So it's the counter current flow ensures that oxygen always moves from a high concentration to a low concentration from the water into the blood across the length of the gill plate. What this means is the concentration gradient is maintained across the length of the gills. And this is the result. If you had it in a graph form, the blood starts over here at the end of the gill plate, the water starts at the start, the water's full of oxygen, the blood comes in. Even this water that has given up all its oxygen, see this water down here, this water is at like 10% oxygen, but the blood that it's next to is at like 5% oxygen, so it's still giving up oxygen to the blood because this is the oxygenated blood coming in here. Of course, once the blood has picked up like 90% oxygen, by that time it's got to that end of the plate, it's meeting water with 100% oxygen that's coming in, and that is called the counter current flow system. Anywho, that's all we gotta know. Yeah, we can get a boom. Boom! Okay. Boom! Oh, I think I got a bit excited there. I gave a double boom. What happened there? Double boom? Jeez! So, in summary, gills have got thin walls, short diffusion pathway, gill filaments and lamellae create the large surface area. Extensive blood supply maintains the concentration gradient. And then we've got the countercurrent flow system. The blood and the water flow in opposite directions across the gill plate, ensuring a steep concentration across the length of the gill plate. Let's do some questiones on the fish. And then I need to look at the fish dissection. And if you're doing OCR, anyone doing OCR? Who's doing OCR? Anyone doing OCR? If you're doing OCR, you've got to know how the how it's ventilated. Remember we talked about the insects with the rhythmic pumping and the wing beating that ventilates their tracheal system. We need to know how the fish ventilates its system. We know how humans do. Hopefully you've memorized that bit with intercostal muscles and the diaphragm, and we change the pressure and the air moves in inhalation. If you're doing OCR, you have to know how fish ventilate their system. So we're going to do that as well. Everyone's AQA. Okay. Awesome. Okay. Identify structures F and G. Now, again, think of the keywords. What can they be? Below is an image of a fish gill. What must F be? What must G be? Yeah, I'm going to go through that anyway, but yeah, some exam boards do need ventilation in bony fish. I don't think AQA do. OCR do. I'll explain it just for the, somebody from OCR might be watching back on the live stream after, so I'll explain it after. But I'm going to go through some questions first. So what do you think structure F and G is? Look, you only know a couple of keywords for the fish, right? You got to guess. Yeah, yeah, yeah. Boom. Well done, Slay Queen. G is lamella. And we, I think, yeah, yeah. Now here's the thing: G is the lamellae, and F is the filament. Yes. Now you can in OCR you can call F the primary lamellae and G the secondary lamellae. Some people like that, primary and secondary, but I prefer gill filament and lamellae. Okay, describe and explain the advantage of the countercurrent flow. So if you had a revision card, this question covers everything you need to know about fish gills and fish respiration, because this is the explain the countercurrent flow. 3 bullet points, that's all you need to know. 3 bullet points for countercurrent flow. Salmon, yeah, what to eat or to like have as a pet? To eat, salmon are pretty cool. Salmon's quite tasty. Yeah. Cod, pretty boring, isn't it? Thank you, Slay Queen. So we want to say the blood and the water flow in opposite directions. Now, what's the advantage to this? This is going to get you 1 mark for saying that. That's the describe. Blood and water go in opposite directions. What does this ensure? This ensures a something concentration gradient, and then we want to say across the entire length of the gills. Maintains a steep concentration gradient. So what we're saying, folks, is if the blood is coming in at the bottom here and the water's coming in from the top, the way they pass each other ensures that there is a steep concentration gradient across the length of the gill, with oxygen moving from the water into the blood from a high conc to low conc. We just want to get that in, all right? So blood and water flow in opposite directions. If you don't know the other 2 points, you need to get these down. So we want to say, yeah, F is the filament, G is the lamellae. Look, except primary lamellae for F. So some people call that primary lamellae and that's secondary, but there we go. Water and blood flow in opposite directions. It maintains a steep concentration gradient or diffusion gradient. Oxygen is always higher in the water than it is in the blood. So oxygen is always moving from water to blood, never the other way around. And then they've got along the length of the whole of the length of the lamellae. Cool. That, look at this question. It's exactly the same. Look, it's like deja vu, right? The thing we're doing past papers, the advantages, you get questions like this. Look, this was a lazy examiner who said, hey, you see that question? I'm just going to do the same thing, right? So all you're doing there is saying, well, gill lamellae, lamellae or gill filaments create a large surface area, and then you could say something like have thin walls, short diffusion pathway, and then look, explain the countercurrent flow. It's going to be identical. I'm going to show you the answers. What I want you to know is that you just got to, once you revise, it's all exam technique. Lamellae, filaments, large surface area, thin walls, short diffusion pathway, water and blood opposite directions. Blood always higher oxygen in the water than the blood. Diffusion gradient maintained. Notice they often say the length of the gill. That's important. It seems to be important in the mark scheme to say that the concentration gradient is maintained across the length of the gill. So oxygen always moves from water to blood. So you might want to get that on a revision card because it just pops up continually, the same things. All right. Couple more questiones on the old fish. Salmon's decent. When I was younger, I must have been poorer. I used to eat sardines on toast. But I wouldn't eat that now. But when I was younger, I used to love sardines on toast. Sardines in chili sauce or something. And then have that on toast. But I don't eat that now. But when I was younger, I used to eat that. I think I must have been poor. Sardines out of a tin as well. Well, there we go. If you have real, when I say real sardines, fresh sardines caught straight from the sea and fried, they're really nice and crispy. Yeah, fish and chips is decent. Fish is pretty good for you as well. It's got lots of good oils. I think it was the fish oil that got rid of rickets in the UK in the early 20th century. I think everybody was having cod liver oil. I think that had something to do with getting rid of rickets. Not that I would take spoons of cod liver oil, but yeah. All right, all right, all right, all right, all right. What do we reckon? Which option is not an adaptation that helps fish increase their gas exchange efficiency? What are we going with? There's a boom for the person with the correct answer. Which one of these does not help the fish in this first question? What do we reckon? What do we reckon? Blood capillaries lie close to the surface of lamellae to minimize the diffusion distance. Blood flows in the capillaries in the same direction. Notice there's blood flows in the capillaries in the same direction as the flow of water over the lamellae to maximize gas exchange. Gill filaments have many lamellae that increase the surface area for gas exchange. Oh, raising and lowering the floor of the buccal cavity. I haven't talked about that. I'll quickly, if you want to know about that, we'll talk about that. Yeah, you're right, guys. B, 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 B. Whoa. Didn't I say? Where's the old boomer? The old boom box. Boom! Okay. Just for the sake of it, guys, just because you might be like, what the hell is this buccal cavity? Okay, let me just zing and tell you what you got to know about the old fish and their breathing system. Okay, so just for those of you that are interested in how the fish gills get ventilated, how do they bring fresh water? They're not bringing air like insects. There's no air in the water that they're getting, right? They're bringing water into the mouth and then out of their gills. The water comes in through the mouth, goes along the gills, and then out the back. And essentially, they change the pressure by opening their mouth. So when they open their mouth, like us breathing in, our ribs move up, our diaphragm flattens, and all of that business. So we decrease the pressure. They open their mouth, and we say the buccal, however you say it, cavity increases in volume. This decreases the pressure, the water pressure, so it's lower than the water pressure outside, and water is sucked into the mouth. Then what happens? The fish shuts its mouth, and that increases the water pressure inside the fish, larger than outside, and forces the water out through the gills, through the flap of skin, which you do need to know if you're in AQA that the flap of skin is called the operculum. And I'm going to show you what this looks like. So let's have a little look at this fish. Nice one, nice one, nice one. Have a look at this fish, right? He's having a little breathe. Check out his gill flap, his operculum. We'll have another little look at that. So check this fish out here. Oh, here he is. Look at his flap there. When that flap opens, that's the water coming out. Actually, there might be a better one here. Let's go to this one here. Okay, check the goldfish. He opens his mouth. That decreases the pressure and the water comes in. And then you see that flap opening there, basically. Okay, cool, 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 cool. So, anyway, that's ventilation of gills if you're really interested. Now, what you do have to know for AQA, final thing. Well, the water, Matilda, there's not air down there, so the water has oxygen dissolved within the water. That not a lot, but it has oxygen dissolved within the water. And so as they take the water in through their mouth. Remember, they don't have lungs to fill up with water and drown, so they're not like humans who, if we went around opening our mouth underwater, all of our trachea and bronchioles and they'd fill up with water and we'd be dead, right? We would no longer be able to carry out respiration. So what the fish does is they open their mouth. By doing this, the buccal cavity lowers, the volume increases, the pressure therefore goes down, and then the water gets sucked in from high to low pressure. Mass transport, basically, a flow of water in, and then the water goes across the gills, and that's where we get all the diffusion of oxygen, the countercurrent flow happening. And then when they get to the back of the gill, the fish closes its mouth, which increases the pressure, and the water is forced out. Now, if you're doing AQA, you do need to, like, what I want you to do is so that if you have a question on a fish dissection and you're like, oh my days, you need to just have 3 bullet points in your head that you can say. Like, so pin the fish to a dissection tray or cutting board, or use a sharp scalpel. cut the operculum off, or open the flap of skin. Those flaps of skin that protect the gills are called the operculum. So pull those back, cut out the gill arch. The gill arch is the bone that all the gill filaments are attached to. So you can cut one of those out, and then you can say, look at the gill filaments, and draw them. Easy as that. Just know 3 things. So before you go in the exam, you just have to know a couple of things that are going to get you the marks. Yeah, so they don't need lungs. They've got loads of mitochondria in their body cells, and they don't need lungs for that, do they? So once their blood is filled with oxygen, their blood is shooting around the body. They have a heart, so the fish heart pumps the blood around the body. Actually, the heart pumps the blood to the gills, picks up oxygen, and then it goes through the capillaries around the body and drops off oxygen on its way. So to the respiring cells, they diffuse through the tissue fluid and out of the capillaries into the respiring cells. So they don't need lungs. Lungs are just our way of bringing air in. Lungs are really just a way of bringing air in to our gas exchange system. Fish bring water in, which has oxygen dissolved in it across their gills, just to get the stuff into their blood. And insects don't have blood. They have hemolymph, but really it's just for carrying glucose and amino acids. Insects bring air in and allow it to just diffuse directly into cells, so they don't need blood to transport it around. Because they're so small, they can do simple diffusion just directly into their cells. Coolio. Right, we are going to finish in a secondo because we have done basically the hour. Let us yeah, so I don't know what people want me to go over next time, but yeah, today's was on insects and fish, and that's all you got to know really for insects and fish. So you should be able to smash it if you get a question on insects and fish. Any topics in particular that people want, and also, do people prefer the 6 p.m. time rather than 7 p.m.? So yeah, it's a couple of questions. Oh no, insects don't take in water. Insects just take in the air by vibrating their abdomen. They force air through the little holes called the spiracles. They create pressure gradients. Basically, they increase the air pressure outside the spiracle, so it then moves in from high to low. The air gets sucked into the spiracles. They don't have water going into those tubes. They want those. The tubes are mainly empty. They're not. There is a little bit of a fluid in the end of the tracheoles, but that's about it. So the air actually just diffuses through empty tubes down and directly into cells with the insect. So over here, look, with the insect, the oxygen, it's not really just oxygen, is it? It's nitrogen, oxygen, carbon dioxide, whatever else is in the air, but they want the oxygen. That goes down a concentration gradient. Why? Because these cells are using oxygen in oxidative phosphorylation to make ATP. So they're using oxygen as the final electron acceptor, and that's what the oxygen is actually needed for aerobic respiration. So it just makes its way down and then goes directly into the mitochondria, where it's used in oxidative phosphorylation. That's the difference. They just can suck in air and it can just diffuse from high to low. Why is it low here? Because they're using the oxygen as the final electron acceptor there in respiration, combining it with hydrogen to make water. Now, in fish, same thing. They want the oxygen as the final electron acceptor in oxidative phosphorylation, but fish are underwater, so they're bringing water in and they're taking air that is dissolved in the water out. But of course, insects live on the land, and they've got a plentiful supply of air around them. Oh, cool, cool, cool. Yes. Do you think 7 p.m. is better than 6? Today was 6. We can go through any year 13 topic. Yeah, we can go through Krebs cycle. Yeah, you're right, Mathilde. So fish take in water which contains dissolved oxygen, which enters the cells. They respire. Then the CO2. Yeah, the CO2 is carried back round to the gills and will diffuse from a high to low. Yeah, you got the same thing, and it's going to diffuse out into the water that's crossing the gill plates. You're right. Yeah, you're right. So that's how they extinguish. They're still doing gas exchange. They just don't focus on that in the exam because the countercurrent flow is so complicated before you bring in carbon dioxide as well. It confused people, but you're absolutely right. So the carbon dioxide, for the same reason, will be diffusing the opposite way. Yeah, I don't mind either one, 6 or 7. Anyway, I'll put a post up and see which topics you guys want to cover for next week. All right, guys, take care, and I will see you next week. Just keep up with the exam practice, physics and maths tutor, save my exams, all that stuff. Use your CGP, make revision cards. Remember, most of A-levels is a memory job and consistent. You got to work from that. If you work from now till your exams, you'll smash it. You just got to keep on. All right. You guys have been amazing. I will see you next time.
I've had training.
The training is nothing. The will is everything. The will to act. Yield.
You're still here? It's over.